All right, looks like we're good to go.
Okay, sorry, I just had to unmute. So 1 second, let me my clicker. So thank you, Dr. Knight, for the introduction. It's really an honor to be speaking to you all. I've managed to catch a couple of the previous talks, and really wonderful speakers have come before me. So it's really an honor, and thank you for sharing your time today. So I'm going to talk about peripheral drivers of Alzheimer's disease and cerebral small vessel disease that we've identified using what I'm calling proteogenetics. All right. So here are my disclosures, really just intramural research program funding. I'm a intramural research program investigator at NIA. And yeah, so the basis of really what I'm going to talk about today is based on this framework that systemic health conditions, physiological factors, things that lie outside the central nervous system really can increase risk for Alzheimer's disease and increase risk for dementia. And there's been considerable epidemiological work, considerable translational research demonstrating that these risk factors, like things listed here, like diabetes, hypertension, systemic inflammation and autoimmune conditions, they can sort of increase risk for future dementia. And this figure on the right is something that was published by the Lancet Commission not too long ago. And the main point that I want to emphasize here is that about 30 to 40% of the attributable risk for dementia was estimated to be due to these comorbid conditions, that many of which lie outside the central nervous system. So it's our hypothesis that plasma proteins might mediate the relationship between some of these comorbid conditions, some of these physiological factors that lie outside the CNS and Alzheimer's disease and dementia risk, right? And so the framework is really outlined here. We think that clinical disease, even subclinical disease and normal age-related processes can lead to abnormal protein expression or changes in protein expression across various tissues. And when these proteins are secreted into the bloodstream, they can, through various conduits, influence cells within the brain. So we outlined some of the, or at least previously, a number of these proteins in plasma have been sort of identified and linked to a number of target cells and known effects on these cells. But we think there's many more systemic proteins out there that are involved in Alzheimer's disease and dementia risk that really have yet to be discovered. So a lot of the work up until now is really focused on using targeted protein assays, looking at proteins one at a time and relating them to dementia risk, to MRI related outcomes, to whatever you like. And this has really been based on sort of this hypothesis, or hypothesis generated, hypothesis driven approaches. And largely this is, it's also been based on proteins of convenience, like the availability of C-reactive protein, for example, in the cohort. And so we get, a lot of studies focusing on a very small subset of proteins. Now, with the advent of proteomic platforms like Soma, Logic, like Olink, that are now available, which allow for the measurement of thousands of proteins with a small amount of sample, we can now do these types of analysis in a data-driven, sort of untargeted, unbiased way, and really start to capture the proteomic signature of various adverse neurocognitive outcomes. And we've done this recently in a paper that was published in Nature Aging, where we essentially did a large-scale proteomic analysis of dementia risk, so of future dementia incidents in people who were non-demented at the time of protein measurement. And so I'm going to talk a little bit about some of the main findings from this paper, and then spend most of today talking about some of the ongoing research, which I find even more interesting and more exciting. So the main takeaway from the paper that we did initially, in which we performed the proteome-wide association study for instant dementia, is illustrated here. So we measured 5,000 proteins in almost 5,000 people in the ERIC study. So the ERIC study is the atherosclerosis risk and community study. It's a multi-site, a community-based cohort within the United States. But we were able to measure these proteins in a set of people who are non-demented and ask, how does each protein predict future dementia risk over a 5-year follow-up period? And so, just like a GWAS, we did one protein at a time, and we adjusted for the comorbid conditions listed down here because we were interested in proteins that showed an independent association with dementia. And we found about 40 proteins that were associated with dementia risk using this approach. So we have 40, most of these are novelly associated with dementia, novelly associated with Alzheimer's disease, haven't really been described previously in the literature. So we have, just like that, we have 40 new potential biomarkers. However, we wanted to take it a step further. And what we did is really ask the question, are any of these proteins causally implicated in Alzheimer's disease? So, we used an approach called Mendelian randomization to look at the causal relationship between proteins and Alzheimer's disease. And I'll talk about this a little bit more later in the presentation. But as part of this analysis, we really identified 2 proteins, SVEP1 and angiostatin, that were associated with Alzheimer's disease. I could say was causally actually associated with Alzheimer's disease based on this Mendelian randomization approach. So we think these 2 proteins are mechanistically relevant. SVEP1, really hasn't been well characterized, but it's thought to facilitate cellular adhesion to the endothelium in the context of inflammation. And then angiostatin, we do know more about, hasn't been characterized in neurodegeneration or Alzheimer's disease, but is a drug target for cancer treatments. So we have 2 new potential therapeutic targets for Alzheimer's disease now that may be targetable in plasma, and we have genetic evidence to support this, which increases the likelihood that the therapeutic target may pan out. So one of the takeaways was that we identified this immune protein network, this cluster of proteins surrounding NF-kappa B that are regulated by this pro-inflammatory transcription factor that actually are associated with reduced risk for dementia over a 5-year follow-up period. So, as you can see, we see proteins like TREM2 or IL-18, which are known to be expressed by microglia. So we think this is sort of a proxy measure of microglia phagocytic activity, and we see that higher levels is protective. So with this study and a number of other large-scale proteomic analysis of Alzheimer's disease that were published around the same time, they really yielded some really good insights, but I think one of the limitations was that they focused on older adults. So we know that Alzheimer's disease and other forms of dementia really do have a protracted preclinical period. So pathology really starts to begin to develop on the brain in 1, 2, or even more decades before the onset of dementia, before the onset of mild cognitive impairment. And it's pretty well understood, right, and agreed upon at this point that the pathogenesis of Alzheimer's disease really begins in middle adulthood. And this is the same for other forms of, other non-Alzheimer's forms of dementia, like vascular dementia, etc. So, and that's really illustrated nicely illustrated in this cartoon down here. So if pathology begins during middle adulthood, it's important in our proteomic analysis, if we really want to understand the biology that's associated with development of disease, to really begin to assess the proteome when disease starts. And that was really the objective of the current work. We wanted to conduct a midlife plasma proteomic analysis of 25-year dementia risk. And the key point here is that we're measuring proteins during middle adulthood and relating them to future dementia over multiple decades. So the first objective was really to understand the early peripheral biological changes associated with Alzheimer's disease, associated with dementia risk. And then secondly, to identify novel plasma biomarkers during middle adulthood that could be used to stratify people according to the risk for Alzheimer's disease, dementia, and other disease relevant in the phenotypes. So we used the ERIC study again to do this. This time we looked in an earlier wave of the cohort away from people who were closer to middle age. So this analysis, we started with a discovery analysis, just trying to identify dementia-associated proteins. And we did this in about 11,000 people who were 60 years of age on average, most range between 55 and 65. And in this 11,000 people, we measured, again, these 5,000 proteins. So we were able to relate proteins one by one to dementia risk over a 25-year period. And over this period, there was about 1,900 incident dementia cases identified. So in addition to relating proteins to full-term dementia risk over this full follow-up period, we looked at proteins in relation to dementia occurring within 15 years and the relationship between proteins and dementia occurring beyond 15 years in our near-term and long-term analysis. And I'll talk about why we did that in a second. So here's the first real big takeaway from our proteome-wide analysis. This is a full-term dementia risk over this 25-year follow-up period. So we see these proteins that are in sort of darker green blue here. These proteins, when measured during middle adulthood, are significantly associated with dementia risk at this Bonferroni corrected significance level. So after adjusting for multiple comparisons. And this is with adjustment for demographic factors, cardiovascular risk factors, and kidney function. So some important potential confounders. But all in all, we have 26 dementia-associated proteins, and we see GDF15, one of our proteins, which rises well above the rest. So the kind of the ApoE4 variant of this proteome-wide analysis. So we also, well, what we did next after identifying those 26 proteins was we asked the question, well, are any of these midlife proteins associated with near-term dementia risk? And when I say near-term, again, I mean dementia occurring within a 15-year period after protein measurement. And as you can see here, when we looked at near-term dementia risk, we identified 7 proteins that were associated with incident dementia. GDF15 you see is displayed again, but we also had these other 6 proteins that were novel, at least to our analysis. So they weren't identified when we looked at full-term dementia risk. So, here we have these proteins that seem to be associated with dementia maybe later in the disease process, closer to the onset of dementia itself. And we can say that they're probably come online, they're more relevant when pathology or a certain level of pathology already is present. And if the are indeed causal, and that's a big if, these proteins are more likely to accelerate the disease more than necessarily trigger it. So we also looked at midlife proteins in relation to long-term dementia risk. And so that's dementia occurring beyond a 15-year follow-up period. And really we see GDF15 identified again, but in addition to that, we see 6 others. And these 6 others have been identified in our full-term analysis. So they were previously identified. And so not necessarily novel to us, but what we can say about these 7 proteins is that differences or abnormalities in expression occurring 20 or more years before the onset of dementia seem to be relevant to dementia risk. And we can extrapolate from that these proteins might be associated or implicated in some of the earliest features linked to neurodegeneration, and whether that be amyloidosis, vascular dysfunction, or sort of immune dysfunction. And so we'll talk about the characterization of protein biology in a second. So I'm going to just breeze through this graph, leave it here for you all to look at. We characterize all the proteins in relation to their near-term and long-term risk of dementia. But I really want to focus on this graph on the right because we are suggesting that we have a number of potential midlife biomarkers for dementia. But our cohort is arguably in the later midlife period with a mean age of 60. So what we did next was we asked, well, if we look at people just in their 40s and 50s, so the lower half of the cohort, if we just look at them in isolation, do we see similar levels of association between proteins and dementia risk? And the answer is yes, for the most part. We see the hazard ratios of the primary analysis and the younger analysis compared to each other here. And Many of them are comparable, and even for some proteins like NDST1, we're seeing a much stronger association when we look at our 40 and 50-year-olds. So I do think we have some potentially bona fide midlife biomarkers for dementia risk. So all in all, we identified 32 dementia-associated proteins, and here's how they sort of are divided based on the relationship with dementia across various timeframes. I'll note that those that are starred, those 4 proteins that are starred, have been already nominated as their potential therapeutic targets for Alzheimer's disease. And this is through the Accelerated Medicine Partnership for Alzheimer's disease, which some of you may be familiar with, but this is a very nice consortium which focuses on proteomics of brain tissue. And these proteins have been nominated through those types of analysis. So it's good to see some proteins that are nominated based on brain tissue analysis also come up as relevant in midlife in plasma, potentially decades before the onset of dementia. So we also then asked the question, what do these proteins represent? Like, what aspects of biology are they involved in? And they kind of fall into 6 major categories. Most of them fall into 1 of 4 categories. So immune function, proteostasis or autophagy, a synaptic function, and then a lot of the proteins are involved in extracellular matrix organization. And then we have a fewer involved in vascular function and metabolism. But they sort of span the gamut of these processes, but some processes are enriched more than others. Let's just say that. And we can actually look at this in a more formally using an enrichment analysis or gene set enrichment analysis. So we did this using ingenuity pathway analysis. We essentially lowered the threshold and looked at the top several hundred dementia-associated proteins for full-term dementia risk, near-term, and long-term dementia risk. And we looked at the overlap between this set of proteins and proteins known to exist in different biologic pathways. And the bars on each of these figures expresses the degree of overlap. All the bars above the line, dotted line here, are significant after correction for multiple comparisons. And then we have the z-scores for direction of activation in white here. So if we look at biologic pathways that are relevant to near-term dementia risk, we see some interesting pathways, most of which are almost uniformly activated or predicted to be activated. But some of my favorites, are platelet-derived growth factor, which just seems to be strongly activated. This, platelet-derived growth factor beta is a biomarker for blood-brain barrier function, parasite injury. And so it's interesting that this is coming up here. Also, we see interleukin-3 and JAK-STAT signaling. So 2 immune pathways that are strongly activated as well. But the story is a little different, or a lot different, I should say, when we look at proteins or pathways associated with long-term dementia risk. We see different processes. We see matrix metalloproteases, we see proteostasis function, bile acid, biosynthesis, so liver function, and then some more immune processes, but from a different perspective. So it's granulocyte adhesion and diapedesis here. So the major takeaway is that the biology differs as we get closer to the onset of dementia. So what we did next was we used, working with collaborators, we attempted to replicate some of our dementia protein associations, but also use their complementary data sets to learn more about where these proteins exist in the various biology of neurodegenerative disease. So, we worked with the 3 cohorts listed here, and I'm just going to talk about some of these sort of replication analysis. And one of the important ones was done in collaboration with the group at Oxford using the European Medical Information Framework for Alzheimer's disease. But they have this nice cross-sectional cohort of older adults who have been diagnosed clinically with either as being cognitively normal, having MCI, or having Alzheimer's disease. And so, this is not an exact replication, but from this, we can really ask the question, are our proteins sort of Alzheimer's disease specific? Because in the discovery analysis, we're focused on all-cause dementia, which doesn't denote a specific etiology. But if you can see in this heat map here, the first column, a number of the proteins that are measured. So if they're in gray, they just weren't measured in this cohort, but a number of the proteins that were measured were significantly associated with clinically defined Alzheimer's disease. And this was good to see, reassuring for our analysis. And then we saw a number of proteins that were also associated with progression from mild cognitive impairment to dementia, Alzheimer's type dementia, and suggesting that these proteins are relevant, again later in the symptomatic progression to dementia as well. So that was the first sort of replication or pseudo replication that gave us more confidence about the proteins identified in our discovery analysis. So we then were able to work with Whitehall 2, which is a very nice cohort in the United Kingdom. And what was neat about this cohort is they also had the same proteins measured during middle adulthood. Okay, so they have a group of about 2,000 people, after the exclusions, about 1,300 for us. But this group of people in their mid-50s, and they have this nice follow-up data. So we could relate all of our proteins that we identified in the primary analysis to risk for cognitive decline over multiple decades in this cohort. And we see that a subset of our proteins, like our heat shock protein and matrix metalloproteinase and then GDF15, they all are associated with greater rates of cognitive decline when measured in middle adulthood. So a subset of our proteins seem to be associated with some of the earliest symptomatic progression that may preempt actual progression to dementia. And we do note that there is this power issue here, which probably made it difficult for us to find more associations. But this actually did provide some additional data for at least a few of our proteins. And then what we did next was we were able to relate use data from the European Medical Information Framework to understand more about how our proteins were associated with neurobiology, specific neurobiologic processes. And we're only able to do this because this cohort has these same plasma biomarkers measured. Also concurrently, they measure cerebrospinal fluid. And so we can relate individual proteins to amyloid, to tau, and other neurodegenerative biomarkers that I'm going to talk about. So as you see here in the proteins that were measured, a number of them were associated with amyloid positive status, with phosphorylated tau at the p-tau 181 specifically. And so these are proteins that are seem to be associated with Alzheimer's pathology, right? We have amyloid and p-tau, sort of the hallmarks of Alzheimer's disease. And so we could say, okay, these proteins maybe not specific to Alzheimer's disease, but they're sensitive to Alzheimer's disease. And then we have total tau, which is just a general neurodegenerative biomarker. But we can go beyond that, and with the measurement of neurofilament light, which is a measure of just pan neuronal loss, neurodegeneration, we see a number of other proteins that are implicated. And we can say that, okay, for proteins like MMP12, which is not associated with amyloid or p-tau, but associated with NFL, this is maybe this protein is associated with neurodegeneration, but not specifically Alzheimer's type processes. So you can really begin to understand where these proteins fit in terms of their relationship with aspects of neurobiology and specific neurodegenerative conditions. A YKL-40 is our neuroinflammatory marker, and probably the one I was most excited about seeing, given the interest in neuroinflammation. And we do see a number of our proteins associated with YKL-40. I'm going to just point out GDF15 here, which seems to be exclusively associated with YKL-40, again, our neuroinflammation biomarker, but not, okay, so GDF15, right, it was our top clinically associated biomarkers, strongly associated with dementia risk in our cohort at various time frames, associated with cognitive decline in Whitehall 2 study, associated with Alzheimer's disease clinically defined in the Oxford cohort, but not associated with amyloid positive status and not associated with phosphorylated tau. So this is really interesting. It's, a strong biomarker for clinical symptoms, certainly, but doesn't seem to be related at all to amyloid or p-tau, but exclusively to our marker of neuroinflammation. So GDF15 seems not to be Alzheimer's specific at all. So, just something that we're going to keep our eye on. But I'll move on from that and talk about how we use some gene expression and protein expression data across various tissues to get a sense of where these proteins were emerging from, where they've been given off from, to learn a little bit more about their place in neurodegenerative disease biology. So we use GTEx, gene tissue expression, and human proteomics to do those things. And here just more heat maps of the results for each protein, the degree of expression across various tissues. So we have gene expression on the left, protein expression on the right, and just some major takeaways. I know there's a lot of data here, but there's 3 major points, right? So we have among our dementia-associated proteins, we have a set of proteins that are just expressed at the gene level or the mRNA level and at the protein level across various tissues and don't seem specific at all. Then we have a set of proteins like complexin 2, like complexin 1 and cerebellin 4 that seem to be specific to the brain. They're almost exclusively found in brain tissue. And this makes them really good biomarkers because what we are picking up on in plasma is then likely to be something that's emerging from the brain, and we can be very confident of that given their it seemed to be exclusively expressed within the central nervous system. So then we have the third category of proteins, which is not expressed in brain tissue at all, and that includes a lot of our dementia-associated proteins, even GDF15, which was most strongly associated with dementia risk and has been replicated, but it's nowhere to be seen when we look at brain tissue, both at the protein and mRNA level. So for those proteins that were expressed in brain tissue, we can ask, okay, are they differentially expressed in Alzheimer's brains? And we were able to use data from consensus cohorts of brain tissue proteomic analysis that are available to do this. So we see for those that were measured, we see differential expression of a number of our dementia-associated proteins that we identified in plasma. And now, again, this is in brain tissue. We see a lot of our synaptic proteins being downregulated in Alzheimer's brains. And we see a number of our stress response, heat shock proteins, for example, being upregulated. So 2 takeaways: our midlife dementia-associated proteins that we identified in plasma seem to be enriched for proteins that are known to be abnormal in Alzheimer's disease brain tissue. So there's a more overlap between the 2 than what we'd expect by chance. And the fact that these proteins are altered both in brain tissue of Alzheimer's disease patients and in plasma well before the onset of disease does provide additional support for their mechanistic relevance. And at the very least, they make potentially really good candidates as plasma biomarkers. So I'm going to give the heat maps a break for a second and talk about our network analysis. I've shown pathway analysis previously, but we were able to make some really good insights using a network-based approach. So we use an approach called NetBoost, which is quite like WGCNA that some of you may be familiar with. So this is a data-driven protein. We used it for protein clustering, and essentially we were able to identify modules or clusters of co-expressed or very highly correlated proteins. And using this approach, for the full set of 5,000 proteins measured during midlife, we were able to identify 20 protein modules. Okay, so we extracted each module and they ranged in size from 23 proteins to almost 700. And this dendrogram down here shows the protein specific categorization. So that's 5,000 proteins down there. So we identified, person specific, we quantified person specific module expression. And we were then able to relate each module to dementia risk. And that's what we did, and that's what is shown here. Using the same models that we use for individual proteins, we related all of our 20 modules to dementia risk over this full follow-up period and found 3, module 1, 5, and 19 that were associated with increased dementia risk over this time. We did the same thing, same modules, but this time looking at near-term dementia risk and found a different module, module 9, that was associated with dementia within 15 years. And then we did the same thing for long-term dementia risk and found sort of a repeat of what we saw when we looked at full-term dementia risk. So all in all, we have these 4 modules, 4 protein modules from proteins and plasma that are associated with incident dementia risk when measured during middle adulthood. So of course, what we did next was ask, well, what do these modules represent? The idea is that the networks, the modules identified by this network analysis represent aspects of biology. Excuse me. So yeah, we use functional annotation tools to really get a sense of what biology these modules represent. And for module 1, which was the largest, made up of 700 proteins about, we see it's enriched for numerous pathways, including some that are more specific than others, but I'll just outline some of them. We see JAK-STAT signaling, we see T-cell differentiation, toll-like receptor ligation. So in summary, this module is very much enriched for immune proteins, immune-related proteins, some specific immune pathways beyond that. And we then take a look at the hub proteins, which are the proteins that are most central to module expression, and proteins that potentially drive the overall module expression. And we see that at least 2 of the hub proteins are known to be differentially expressed in Alzheimer's disease brain tissue. Okay, so we did the same thing for module 5, very different biology is implicated here. We see extracellular matrix organization or disorganization more emphasized among the proteins that are known to exist in this module, but with some evidence of immunologic relevance as well. So module 5 takes on sort of a different role in biology, it seems, and 4 out of 6 of the hub proteins that we see are differentially expressed in Alzheimer's disease brain tissue. So it's even more enriched. And then we see for module 19 down here, which was most strongly associated with dementia risk. Again, it's an immune module, less specific in terms of what actually the role is in the various immune pathways. So we see MAP kinase signaling. We also see this longevity regulating pathway, which is very interesting. We plan on looking into that. But like one of the takeaways, if we look at the hub proteins, we see heat shock protein, and another heat shock protein. So it's enriched for immune proteins, but also, these proteins that are known to be involved in proteostasis and stress response. And many of these proteins, all those with asterisks next to them, are known to be differentially expressed in Alzheimer's disease brain tissue. So this is the most enriched module we've seen, and I'm really excited about what this might represent in terms of underlying biology. Then module 9 was the one that was associated with dementia in the near term, again, within 15 years. And we see very different biology here. We see complement and coagulation being the things that are very much enriched and strongly enriched, I'll add. And then we see that a lot of these hub proteins are, again, differentially expressed in 80 brains. So if we look across the time course leading up to dementia onset, we see this multi-decade evolving, let's call it an immunologic signature. We go from involvement of JAK-STAT, namely downregulation of JAK-STAT and T-cell differentiation. At the same time, we see upregulation of these other aspects of immunity. And as we get closer to the onset of dementia, we're seeing more involvement, complement, coagulation, and these other processes as well. This is from our network analysis, and this is what we've taken away from the canonical pathway analysis that I've shown previously. So we can sort of learn about the evolving, at least peripheral biology that precedes dementia risk in this way. So one of the questions we had next was, well, are these proteins and protein modules, are they mechanistically relevant? Do they play a causal role, or are they just sort of bystanders, or do they play some sort of compensatory role? And we were able to begin to address this using what we call two-sample Mendelian randomization approach. So essentially, for each of the dementia-associated proteins, we ran a genome-wide association study on plasma protein level. And we did this with the midlife proteins measured in the Enriched study. So we took the variants associated with plasma protein level, which we're calling PQTLs, and then we asked, are these PQTLs associated with Alzheimer's disease? And we were able to look at the overlap between the protein-associated variants, or PQTLs, and their association with Alzheimer's disease using a second GWAS study, or at least a summary statistic from the second GWAS study. This is something that's been published by Kunkel and colleagues a few years ago. But so this is how we get to this two-sample Mendelian randomization. And I know that was a very oversimplified explanation, but Mendelian randomization is a great tool because given some assumptions, and the known sort of random allocation of alleles throughout the population, we can make causal inferences about protein Alzheimer's disease associations, and circumvent some issues related to reverse causation and confounding that are inherent in most sort of epidemiologic research. So with all that said, among the 32 proteins, we found 2 that seem to have some evidence, at least in this inverse variance weighted approach, for an association with Alzheimer's disease. And SERPINA3, this one in red here, was actually supported in the various sensitivity analysis. So we're excited about that, potentially causal. We didn't see anything with the modules, but we also performed this Mendelian randomization approach in the backwards direction. So we're asking, is Alzheimer's disease, is genetic propensity for Alzheimer's disease, is it associated with abnormal expression of protein level? And we're talking about midlife protein level. And we see based on the red in this table here that we see many more associations and much stronger associations in this reverse direction. And what this suggests is that Alzheimer's disease biology leads to protein changes in middle adulthood. All right, and we see a little bit of evidence for the other way around, but it's much more stronger in this direction for a different set of proteins. And this is even the case for Alzheimer's biology changing proteins in middle adulthood, even proteins like GDF15, which aren't expressed in brain tissue. So just to summarize the Mendelian randomization results, we see some support for a mechanistic role for SERPINA3 in Alzheimer's disease. And this is something that has been nominated previously as a therapeutic target, and it's been genetically implicated in other studies. So we're excited about pursuing this target with more mechanistic studies. But another big takeaway is that the large proportion of dementia-associated proteins that we identified during midlife seem to be abnormal as a result of biologic changes accompanying Alzheimer's disease. So they may play a compensatory role. It's not really clear. And I will say that a lot of these proteins, what we do have now for a lot of these proteins is genetic validation for their role and potential role as biomarkers for Alzheimer's disease specifically. And although I didn't show this, but we were able to look at the using Mendelian randomization, look at how these dementia-associated proteins were related to other traits and phenotypes. And we found that a lot of the dementia-associated proteins were causally implicated in vascular, inflammatory, and metabolic conditions that are known dementia risk factors. So they may operate, if they are causal, through this indirect pathway by causing some of these comorbid conditions, which we know to be dementia risk factors. So here we have this new set of plasma biomarkers for Alzheimer's disease and dementia that are linked to various biologic pathways, things like proteostasis and immunity, which are known, widely known to have some role in Alzheimer's disease, some role in various other neurodegenerative conditions. And now we have biomarkers that as early as 40 and 50s seem to be perturbed, and that may allow us to capture this heterogeneous contributions to neurodegeneration. And here these squares represent, these are just some of the prioritized ones from our study, and the squares represent the level of evidence. So that's for, I showed proteostasis and immunity. These are synaptic biomarkers, and then a number that were involved in extracellular matrix organization and vascular function. So just some takeaways to summarize, during midlife, there seems to be a plasma proteomic signature associated with dementia risk. That's for certain. And we can say that the top proteins associated with dementia risk at midlife do fall into about 4 to 6 overlapping biological categories listed here. So the immune protein networks do seem to be altered at all stages of this disease process from 25 years till dementia onset. Onward, this immunologic signature does seem to change with time, and many of the dementia-associated proteins causally linked to Alzheimer's disease risk factors like cardiovascular disease and inflammatory conditions. But then, as I mentioned, a large proportion of these proteins seem to be altered as a result of dementia risk or Alzheimer's disease specifically as well. So, now I'm going to switch gears and really talk about how we've used a similar approach to understand the peripheral biology of cerebral small vessel disease. And this is a project that's being led by one of my trainees, Gabriela Gomez here, who's a third year medical student. But what the question that we're asking here is, can we use this plasma proteomic approach to understand, what's driving some of these various forms of cerebral small vessel disease, and can we identify novel biomarkers for each of these conditions? Now, these conditions are relevant because they mean something clinically. Having cerebral small vessel disease, for example, has been associated with cognitive decline, dementia risk, and some of these other phenotypes listed here. So it's a target of potential treatment, and it's something that's gaining increasing. People are increasingly aware of, given initiatives like the vascular cognitive impairment and dementia type studies. So we looked at white matter hyperintensities, lacunar infarcts, and cerebral microhemorrhages. So we use protein data from a later wave of the ERIC cohort to do this. And we looked at a subset of 1,500 people who had these proteins measured and concurrently had also undergone 3T MRI. So in this way, we were able to relate all of our 5,000 proteins to each of these small vessel disease phenotypes. And as you can see in this panel A, we did identify a number of proteins that were associated at a protein-wide significance level with white matter hyperintensity volume. All right, so 13 in all. And we also, again, looked at lacunar infarcts, we looked at cerebral microhemorrhages in the lobar region, and also microhemorrhages in the subcortical region. But we didn't identify any that met significance after adjusting for multiple comparisons for these other small vessel disease phenotypes. But of the 13 that were associated with white matter hyperintensities, a number of them were concurrently associated with, at a more nominal level, these other phenotypes like transgelin here, which seems to be associated with all small vessel disease phenotypes that we measured, whereas some are more specific to white matter hyperintensity volume. So we were able to perform pathway analysis on these sets of proteins too, looking at a larger set at a threshold of like p less than 0.01, for example. And again, we see different biology implicated for each form of cerebral small vessel disease. So for white matter hyperintensities, we see, from this plasma proteomic perspective, we see involvement in cholesterol metabolism, coagulation pathways, and this interesting SPINK1 pathway, which does have some relevance to hypertension. So some face validity there. And lacunar infarcts looks different from a biologic perspective, given these enriched pathway analysis. We see NF-kappa B signaling. This is a inflammatory transcription factor. We see acute phase response and IL-6 just below the threshold as well. So the signature for lacunar infarct seems more pro-inflammatory than does white matter hyperintensity volume. Same goes for subcortical microhemorrhages and lobar microhemorrhages. I'll say for lobar microhemorrhages, this is what we might expect to see. Coagulation and prothrombin pathways are predicted to be activated in those who go on to have these microhemorrhages in their brain. So yeah, the biology is distinct, and oftentimes small vessel disease is sort of lumped together in a category. So I think this evidence that they may have differing etiologies is you know, particularly interesting. We also use this clustering approach to understand small vessel disease. So looking at the 5,000 proteins measured in this group of 75-year-olds, we were able to identify 15 protein modules, and the clustering is demonstrated here. Seven of these protein modules were themselves associated with cerebral small vessel disease. And you can see as here in this heat map, I know the modules don't mean much now, but I'll point out that module 9 and module 10 were strongly associated with white matter hyperintensity volume. And so we looked further into what those modules might represent, same sort of enrichment analysis. We see module 10, which seems to be associated with each form of small vessel disease, is enriched for proteins involved in cell development or the inhibition of cell development, osteoblast signaling, and then IL-11 signaling as well. Whereas module 9 was much different. This was exclusively associated with white matter hyperintensity volume, and we see different biologic processes being implicated here. A lot of more metabolic type processes. So differing biology. We applied the Mendelian randomization approach here as well. We used a GWAS study from that's been published on white matter hyperintensity volume as sort of to define or determine the white matter hyperintensity volume summary statistics. But looking at the overlap between, again, pQTLs in the Enriched study and variants associated with white matter hyperintensities in this other study, we again found some interesting results. None of the identified proteins proved to have a causal relationship with white matter hyperintensity volume using this approach, but we do see that one of our modules, module 3, for which we found numerous pQTLs, was mechanistically or causally associated with white matter hyperintensity volume in this analysis. So we went back and said, okay, what does module 3 represent? And we see, this is just in the sort of epidemiologic cohort, we see that module 3 is associated at a nominal level with white matter hyperintensity volume. And it's like, okay, so we see some convergence with the Mendelian randomization analysis. And then when we look at what the biology of module 3 is, we see it's very much complement signaling, very much immune activation, and specifically activation of C3 and C5 pathways. So from this, we can we have some evidence to suggest that peripheral activation of complement may be causally relevant in development of white matter hyperintensities, again, a form of cerebral small vessel disease. So this project, is really ongoing, and we're hoping to explore this a little further. But I just wanted to show you all how these dementia proteomics processes can be applied to MRI characteristics or other dementia in the phenotypes. And with that, I'll end. So thanks everyone for listening, for being here, and of course I'd like to thank the collaborators. This is a lot of work, involves a lot of different skills, and this is truly a collaborative effort. So, my mentors, Rebecca Gosman, Joe Koresh, and Susan Resnick especially deserve some thanks, as well as the collaborators across the world who've helped us put this together. Thanks again for listening.
Thank you so much. That was really fascinating. It looks like we have roughly 8 minutes or so for questions. Would anyone like to jump in? You can post a question to the chat, or go ahead and unmute and ask yourself. So I do have a couple of questions myself. So you mentioned that with Mendelian randomization, you can make this kind of causal interpretation. What assumption or what model assumptions are we making that allow for that interpretation?
Yeah, good question. So there's 3 main assumptions for Mendelian randomization, and I won't go into, I won't list them all. Actually, I don't think I could list them all by memory, but we do perform a lot of um, sensitivity analysis, which do check the assumptions. But it really relies on this idea. Let me actually go back to it relies on this idea that the exposure or the risk factor is, so the risk factor in this instance is plasma protein level. And it relies on the idea that plasma protein level is genetically determined, at least to some degree. And that varies by protein, of course. But though the variants that determine plasma protein level are then randomly sort of allocated throughout the population, right? So, you may have a variant that's associated with high levels of ESM1, and I have a variant that's associated with low levels. And the degree to which we get those variants is almost random. And so it uses that assumption to say, okay, well, you have variants that are associated with high levels of ESM1. And if we pull all the variants that we can define, here it is two that predict levels of ESM1, do those variants together, are they also associated with white matter hyperintensity volume? Okay, so if you have a genetic propensity for expressing this protein, then the question is, do you also have a genetic propensity for having high levels of white matter hyperintensity volume? And if that is the case, and assuming that these variants like exclusively are associated with the or are actually and exclusively associated with the risk factor, then we can make the assumption that there is a causal relationship.
Thank you. And it looks like we do have a question from Pablo. He asks, could you comment about future projects related to this field?
Yeah, there's really just so much that is being done and can be done with all this. I mean, just the proteogenetics itself, using the ability to now identify proteins that are causally relevant to various diseases is just, it's been amazing for drug discovery and understanding the mechanistic role of various molecular pathways in diseases. So there's people who are, and some actually recently published work has just done a GWAS on every single protein available in the Proteomics platform. That is like 5,000 proteins, and then identified, so has done a GWAS on 5,000 proteins, identify this whole list of pQTLs, and then applied those to hundreds of phenotypes, really to identify these various protein trait mechanistic associations. And that's really been insightful for various diseases. Here we're choosing to really focus our attention on dementia and Alzheimer's disease. But I think there's a lot more that we can do with proteogenetics. But really beyond that, I see one thing that's I like that's being done with these proteomic platforms is we're using them to understand the relationship between various exposures like physical activity or cognitive stimulation and various outcomes, right? We're able to see how physical activity, for example, changes the proteome, and then understand how proteins that are changed by physical activity can influence an outcome. So in that way, we're able to really start to make the mechanistic links between exposures that we know to be related to various outcomes, and go from just epidemiology to biology. And it's this real neat merging of fields that's become, GWAS really allowed that, and I think proteomics allows it in just the same way.
Thank you. And I think Jean Yu, do you have a question?
Yeah, it's a very good talk and a lot of information packed in this talk.
Thank
so
you.
I learned a lot. Thank you. The one question is, you identify this top, 25 some proteins related to the Alzheimer's, that I just curious to know that, how that the proteins, if any genetics regulation on these proteins are overlapped or highlighted in the large jivas from the AD?
Yeah, so the larger, yeah, so we're not saying there are some, and like one of our proteins, EGFR, epidermal growth factor receptor, yeah, has been highlighted in one of the very recent GWASs, the one that for Alzheimer's disease that implicated like 70 to 100 variants. So we're seeing some overlap, and that's just one example. The other protein, SERPINA3, has been implicated in GWASs, but not consistently. And actually that protein's been implicated in the GWAS for disease progression. So it's not as much as, the overlap is not as much as you might think. Like our top proteins aren't clustering soluble TREM2, ApoE, like we're not seeing that one-to-one correlation. And we know that a lot of the variants that are associated with Alzheimer's disease in these GWASs are sort of like these non-coding intergenic or intronic regions, and might not simply change protein expression in an obvious way on the gene in which they're found. So I think we're offering complementary information here, which, like I said, doesn't overlap one-to-one or much at all in terms of individual molecules with what we're seeing in GWAS. Another point I was going to make. Yeah, if we look at the pathways, though, like from a broader perspective, we see immune, we see metabolism. So some of the same biologic pathways that have been identified in GWAS and transcriptional analysis of AD are implicated here as well, but not the individual genes, proteins.
Oh, okay, thanks. Another question is that the highlighter in your results, the particularly one is the GDF15.
Yeah.
It has strong. I mean, I noticed that in the presentation, this is not, look like it's not specific for the AD, but it's more of neuroinflammatory indicator. But later on, you also showed the proteins in the brain expression is not different between AD and the controls.
For GDF15, it's not even measured. Well, it's not available. So a lot of the proteins that we measured aren't even measurable in brain tissue. So we don't think GDF15 is really expressed at a measurable level. And so that's what the gray indicates.
I see. I'm trying to think is if that's an indicator of neuroinflammatory factors, then how that, yeah.
Yeah, personally, I think it's a GDF15, it's been implicated in cellular senescence. And I think it's a sort of stress response protein that's really mostly peripherally, and it may be an indicator of how sort of peripheral disease can influence the brain, or it may be an indicator of the degree of peripheral disease, whether that be vascular or inflammatory, and that disease that's operating outside the brain then may contribute. And I don't know if it's through GDF15 or if GDF15 is just an indicator of the extent of peripheral disease. If that makes sense.
Yeah, thanks. Yep. Again, thank you. That's a very good talk.
No, thank you for being here.
Great. Well, it looks like we probably need to wrap up because I think you have that following meeting. But we can post any additional questions to the Slack channel, and I'll follow up with the recording as well. And Keenan, if you don't mind sharing a couple journal articles with me, and I'll also post that to the group for people who might want to do some follow-up reading.
Yeah, absolutely. I'll give you my article and some others.
Awesome. Thanks. Well, thank you so much again for such a great talk and for joining us today. Everybody have a great rest of your day.
All right. Thank you.
Bye.